Hello everyone, my name is Hamid Ekhtiyari, delighted to be here. I will be discussing mainly about what neuroscience can teach us for prescribing medications for addiction treatment. So that is the basic idea. I put all my slides in the QR code. So if you scan the QR code, I will provide the QR code at the end as well. So if you scan the QR code, you will have access to the entire slide set. So you will be able to go through the things that we are going to discuss, and hopefully there would be kind of interesting discussions. My lab is mainly focused to use brain stimulation technologies for addiction treatment, and we use different technologies like kind of different non-invasive, invasive technologies, and we try to find the best neurocircuits for addiction treatment. But meanwhile, I'm very interested to see how basically neuroscience could reshape the future of addiction medicine. And I mean, okay, so this is a kind of a 2 volumes of book we had with 37 chapters discussing about how neuroscience could reshape the future of addiction treatment. The entire book is available on the QR code, so you can have the book. And now I accepted to be the editor-in-chief of the entire series for the next 10 years. So I'm gonna basically develop 20 books on neuroscience for psychiatry, discussing about how neuroscience is going to reshape the future of psychiatry. So I will be happy to discuss with those who are interested to be volume editors for that book series. So I don't have any conflict of interest. I just had a kind of a one-page talk for Indivior kind of funding that is presented here, and I will be discussing about off-label use of some of the medications. But before starting the discussion, I'm really interested to kind of start with being grateful of those who basically made this happen for us to have that level of understanding about neuroscience of addiction. And I would be interested to kind of basically starting with 6 major figures in the field. Who knows all those 6 major figures in the field? Okay, of course, Alex knows them. Who else? Okay, who else knows all those? You know, do you want to kind of tell us all those 6 names and introduce yourself?
No, I don't know. I know Dr. Stahl.
Okay, you know Steve Stahl. Okay, who else do you know?
Nora Volkov.
You know Nora Volkov. Who else?
No one.
Okay, who knows the second one on the top? Okay. Yeah, of course, Kathleen, you know everybody, yeah, of course, yeah. But others. Okay, so as you can see here, these are the names: Yasmin Herd, Eric Nessler, Steve Stahl, kind of George Koop, David. Now, these are the really giants of the field. And also, I would like to mention 2 local figures here in Mexico that they have contributed significantly to our understanding about neuroscience of addiction. Our local people here from Mexico. Who knows these 2 figures? Who knows them? What are the names? And you know them. What are the names? Sylvia Cruz and the young guy.
No, I don't know.
You know, who knows the young guy? Eduardo, yeah, so Eduardo Garza Villarreal. So, I mean, we definitely need to appreciate the things that they have done to develop the understanding that we have. And as I'm the first speaker, I would have just one slide discussing about the prevalence of substance use disorder. And do you know what is the percent of substance use disorder, last year substance use disorder in the US? Do you have any estimate? Alex, what is your estimate? Percent?
This includes nicotine and alcohol?
Includes alcohol, not nicotine.
I would say 15%.
15%. Who else has an estimate? Okay, the number 15%. Okay, so 40 million people in the US, they had past year diagnosis of substance use disorder. So it's the number is huge. And we discuss about how we can provide them with treatments. But when we discuss about neuroscience and neuroscience of addiction treatment, everybody would have a kind of a sense about complex circuits that we are going to discuss about, all those, let's say, connections and just some confusions about how those things are working. And it's just not most of the time very, very helpful for people to understand those networks. But I'm gonna be more applied in terms of how our understanding about neuroscience could help us for addiction treatment. And starting from dopamine, okay? We all agree that dopamine is the central core, right? Dopamine is the central core for addiction and addiction treatment. And there are two pathways that are involved with dopamine production and function, mesolimbic and mesocortical networks. And these two networks basically, as we know, they start from VTA, ventral tegmental area. They start from that kind of yellow dot there, and then they project from yellow dot, which is ventral tegmental area, to limbic area. So, and because that ventral tegmental area is located in mid part of your brain, it is called meso. So from meso to limbic area. Limbic area is nucleus accumbens. And then also to cortex. So meso, limbic, and mesocortical, okay? So these are two pathways that we are going to discuss about and see how they are going to inform our treatment process, okay? And then when we discuss about dopamine, we know that dopamine is basically being produced and recycled in presynaptic terminals, okay? And we all heard about that, kind of the process of what we call dopamine receptors, dopamine transporters, vesicular monoamine transporters that basically collect dopamine into the vesicles. And we know that the production is not actually happening in dopaminergic nerves in terms of the cell body, but the production happens in the nerve terminal. Okay, so that is the process. And so the production happens in basically the area that the projection is ending up there, where we have basically the presynaptic terminals. But let's just make it kind of one step ahead in terms of how these parts are working together, okay? So we discussed about ventral tegmental area connected to nucleus accumbens and releasing dopamine in nucleus accumbens, right? So that was the first step. And then there is a significant role for glutamate. So glutamate from the cortex has projections to VTA. So that is the next player that is doing some like a cortico-subcortical regulation. And then we have GABA in VTA. That is the third player, okay? And then we have opioid neurons that are interacting with glutamate and GABA. So that is the main core in terms of how these four major neurotransmitters are interacting with each other, okay? And finally exerting the release of dopamine in nucleus accumbens and also in cortex. As we said, mesolimbic and mesocortical. Okay, so that is the basic structure. There is one more really important neurotransmitter in this system, and that is basically norepinephrine. We know that norepinephrine also interacts with the system. Okay, so that is the structure that we have. So there is a balance between GABA, glutamate, dopamine, opioids, and norepinephrine. And that balance is basically how things are working in the brain. But different substances work on different parts of this network, okay? So when we talk about each substance, we need to discuss how that specific substance is interacting with these basically 5 major neurotransmitters. And then if we are going to discuss about a medication, that medication is targeting these circuits. That medication is targeting basically one of these major players in the circuit. And then whatever we have here is going to be representing in the brain in terms of how different parts of the brain are interacting with each other. And we know that in a healthy situation, we have a good cortico-subcortical inhibition. So that basically red arrow that shows we have a control on our emotions around impulses, right? But when we have an extended activity in nucleus accumbens, as you can see here in this figure, when we have extra activity in nucleus accumbens and places like ventral putamen, areas that are related to nucleus accumbens, because you have lots of dopamine there, then that basically nucleus is driving the brain, and then basically you do not have the inhibitory control, okay? So that is the problem that people with substance use kind of experience in terms of they have lack of control, and that is something that we try to see how we can basically modulate with different medications in the circuit that I discussed about. So whatever we have been discussing, that circuit is going to be eventually end up to this network and help us to have control on using substances. Why do we need to learn about these, what we call biomarkers, the kind of the biological processes that are happening inside the brain? So as an example, one of those biomarkers that we have been discussing about glutamate, right? So we have been discussing about glutamate and how glutamate is basically interacting with subcortical areas, okay? So that is the glutamate neurons that we have, which was interacting with subcortical areas. People showed in different studies that the level of glutamate that you have in your DLPFC is a marker of the chronicity of your substance use. It's also a marker of, if we want to do things like TMS on DLPFC, TMS is going to change the level of glutamate that you have in your DLPFC. And that is one of the mechanisms that you can exert. When you receive TMS on DLPFC, then you have better control on subcortical areas because you change the level of glutamate on that part of the brain, right? So these markers basically inform the way that we are going to treat addiction. And that is the way that neuroscience-informed addiction treatment is going to happen in the future. There are other measures of, let's say, cortico-subcortical connection. So we can measure the connectivity with things like fMRI and how these things are interacting. So that could be another biomarker. Or even we can measure the level of neurotransmitter receptors or neurotransmitter reuptake inhibitor, reuptake kind of transmitters, and that could be another marker. I can imagine you have seen those images of dopamine transporters in methamphetamine users. After a period of time, they have a lower level of dopamine transporter, right? Because of the substance use over time, their dopamine system does not work really well. And that's a diagnostic measure of chronic methamphetamine use, right? So that is an information in terms of how the system works. And we know that basically that measure could also be what we call a monitoring biomarker in terms of showing that over the time, how the process of recovery is going to change the level of dopamine transporters that you have in limbic system, the nucleus accumbens, and how that is a marker of recovery over the time, okay? So being back to this figure, we recently kind of have a paper that is going to be published in Nature Mental Health. I provided a QR code to the paper, and we are discussing about all different biomarkers that could inform the process of addiction treatment in different levels. And when we discuss about biomarker by itself is meaningless. So we shouldn't say biomarker alone. We should say, is it a diagnostic biomarker? Is the monitoring biomarker? Is the response biomarker? Is a safety biomarker? Or even it is a susceptibility biomarker, that those who have this specific level of dopamine in that part of the brain, they would be more susceptible to get addicted when they use substances. So all those different types of biomarkers that we have. And that level of understanding basically is now helping us to have a sense about how addiction is happening over the time and how brain is changing over the time based on these biomarkers. So we know that when we start with addiction, it basically starts with having further mesolimbic dopaminergic activity and engagement of nucleus accumbens, right? So these are the measures that we have been discussing today. But we know over the time, the prefrontal cortex is reshaped because of changes in glutamate that we have been discussing about. So initially, the process starts with just further dopamine. But over the time, the prefrontal system with glutamate is changing. And then when we go further, we have further involvement of amygdala. And now the addiction is getting more and more compulsive in terms of people are not enjoying anymore. They are just basically using that because if they are not going to use, they would have high level of negative feeling and kind of withdrawal, right? So that is the reason that the amygdala is now involved in that process. And we'll discuss how this basically happens in the brain in that process, okay? And these basically biomarkers right now are giving us basically new definitions in recovery process. One of those new definitions is what we call TRA. We are in the psychiatry meeting, so everybody knows about TRD, and we practice psychiatry in kind of treatment-resistant depression or hard-to-treat depression, but the idea of, do we have treatment-refractory addiction? Who believes that we have treatment-refractory addiction? Just bring up your hand. Who believes that we have treatment-refractory addiction? Okay, so it seems that there are many people who agree that we have people who, they have chronic relapses, we have done everything for them. It's like TRD. When you practice TRD, you see people that you have done 20 different antidepressant medication and they are still struggling with depression, right? So the same concept, but to be able to measure that, the question is how we can have biomarkers to show that process. And also another concept that, there are lots of debates about that in terms of, do we have something that we call pre-addiction? Who believes that we have pre-addiction? It's like pre-diabetes. We know that, before getting to diabetes, we have pre-diabetes. So we are getting there. We do not have diabetes, but we have impaired glucose tolerance test. Hemoglobin A1C is just a little bit higher than normal. You have challenges. You are in a pre-diabetic state. Do we have pre-addiction state? Who believes that we have pre-addiction state? Okay, so, and there are lots of debates in terms of should we introduce this concept to the field or not? What are the challenges, the stigma, all those challenges that could be related to this concept. But to be able to have these concepts, we basically need to have a better understanding about that, okay? Now the question is, okay, if we extend our knowledge about addiction neuroscience, how this basically could be helpful for clinical application, okay? So far I spent 19 minutes discussing about some basics, and now I'm gonna discuss for the next 10, 20 minutes discussing about some clinical applications, okay? One of those applications could be how the imbalance between glutamate, GABA, norepinephrine, and opioid could have clinical presentation, and how we can basically, based on the clinical presentation, decide what type of medication that we can prescribe, okay? So that is one step. The other, how basically inflammation and metabolic toxicity happens in the brain in response to using substances, and how we potentially can prevent that or treat that. How dopamine and glutamate could be toxic in the brain, and how we can prevent that toxicity and how we can treat that toxicity. How our new understanding about neuroscience could help us to understand what we call xylazine. Who have heard about xylazine? Xylazine, okay, so that's a big challenge for us in the US. Now it's a global problem, so people are facing that. Synthetic catechins, synthetic cannabinoids, and then Hypso. Who has heard about Hypso? Hypso? No? High potent synthetic opioids. Okay? High potency. Starting with fentanyl and then a large group of high potent synthetic opioids that we will need to discuss about specific neuroscience of these new substances. And also the complexities of partial agonists like buprenorphine and interactions with Hypso. How buprenorphine and our kind of opioid use disorder medications should be optimized in the era of Hypso with all those high potent synthetic opioids. and potentials for what we call biotyping in terms of based on the biomarkers defining who should receive this treatment and who should receive the other type of treatment. And also some new molecular targets that we can think about. Okay, so kind of few clinical examples. So we have been discussing about kind of excitation and inhibition balance and glutamate and GABA and the interaction between these basically 4 major neurotransmitters. So we know that glutamate is an excitatory neurotransmitter and GABA is an inhibitory neurotransmitter. So they interact with each other and then eventually they stimulate the dopaminergic system to produce dopamine, okay? We know that alcohol basically interacts with GABA receptors and basically activates GABA receptors. So in a normal healthy brain, we have a balance between GABA and glutamate, right? So both of them are in a sort of balance, excitation, inhibition. But when we prescribe, when people use alcohol, what happens? That balance is disrupted. Alcohol has 2 major targets in the brain. One is GABA, and the second target is the mu opioid system. So we are discussing about 2 major targets in the brain. Now the main discussion about kind of GABA, Christos and Rama will be discussing about medications, about kind of mu opioid receptors, things like that. But GABA, when we have further alcohol, we have extension in GABA activity. Then glutamate needs to enhance its function to have the balance. Now we have higher level of glutamate activity, higher level of GABA activity. Now they are balanced to each other. But when people stop drinking alcohol and then go to withdrawal, what happens? We have a very active, very strong glutamatergic activity. And that basically has a clinical presentation. And we show that in a measure we call CIWA, right? And norepinephrine is also involved. So when we have higher glutamate, we usually have higher norepinephrine. So we have these 2 neurotransmitters involved. So what are the clinical presentations that we have in CIWA? Agitation, anxiety, auditory disturbances, clouding of sensorium, headache, nausea, paroxysmal sweats, tactile disturbances, tremor, visualism, all those kind of the hallucinations that people can experience during alcohol withdrawal. And people have discussed about alcohol withdrawal syndrome, and you know about delirium tremens. And delirium tremens basically happens because your brain tries to bring down glutamate as much as it can. So it does its best thing to bring down glutamate. But if the glutamate is too strong and brain uses its entire resources after 48 hours and give up, then delirium tremens happens with kind of really strong glutamatergic activity, and people would have kind of lots of challenges in terms of basically going to delirium and all those challenges, seizure, all those challenges that could happen because of having a really strong glutamatergic activity. So the idea is, okay, if we have lots of glutamatergic activity as we have been discussing right now, and this is basically the NMDA receptor, the receptor that glutamate works on that specific receptor. And we know that receptor basically lets Na and also calcium to go inside the brain, okay, inside the cells. And we know when calcium goes more than what it is supposed to go inside the cells, we have what we call at the end of as you can see on the low part of the slide, we have apoptosis. So this is what we call excitotoxicity. When we have lots of glutamate in the brain, that basically kills brain cells. And then basically glutamate toxicity happens. And then the question is, can neuroscience help us bring us idea in terms of how to prevent that entire presentation, okay? Ideas about how we potentially can even block that sort of NMDA receptor for glutamate for a period of time to be able to manage that. The ideas like kind of using things like memantine and kind of blocking that specific receptors, or other things that we can further discuss about. And there are studies that they show something like a 10 milligram TID memantine would act almost the same as diazepam 10 milligram for alcohol withdrawal syndrome, when the alcohol withdrawal syndrome is not that much strong. So in a sort of mild to moderate cases, not severe cases, okay? And I mean, you know, there are lots of other discussions in terms of how potentially memantine could be helpful in some other psychotic disorder. There are, as you know, there are positive and negative results about that, so I'm not gonna go towards that. So basically the idea right now, when we discuss about alcohol withdrawal syndrome, because we have lots of glutamate activity, we want to have GABA to basically to balance that for at least a period of time. So the gold standard to bring up GABA is benzodiazepine. So basically we use benzodiazepine. I'm not going to go to this. I can imagine our colleagues will be discussing about that. But the idea is just using benzodiazepine with different kind of dosing based on the symptom, based on the SIVA symptoms, whether they have more than 8 of SIVA, and then kind of using the different doses of medications. But also, what else could people try other than benzodiazepine? What is the challenge with managing alcohol withdrawal syndrome with benzodiazepine? What is the problem of using benzodiazepine? It works beautifully well, so it is the gold standard. Everybody recommends using that. You cannot continue doing that. Yeah, of course. So it is good for a short time. So you need to basically do it in a really short time and taper it, and just that's it. So you cannot continue doing it. Is there any chance that we can use medications that could be continued for a longer period of time? So there are discussions about things like gabapentin. And we know that gabapentin, the good thing about gabapentin is something that you can continue using that. The challenge with gabapentin, it is not probably the best thing for when you have the alcohol withdrawal syndrome in the severe cases. And there are discussions, I can imagine our speakers, we will discuss about that. I mean, there are people who love gabapentin, there are people who hate gabapentin. So we'll discuss about that. But the idea of basically using gabapentin, and how gabapentin works. And you know gabapentin is really close to GABA in terms of the structure, right? So the structure of gabapentin and pregabalin is almost really close to GABA. But interestingly, it doesn't work on GABA receptor. So it is not like benzodiazepine. So it does not go and basically interact with GABA receptor. What is the mechanism of action of glutamate? How basically gabapentin and pregabalin work? What is the molecular target in the brain? Calcium channels. Okay. So we are talking about VSCC. Who knows VSCC? Voltage sensitive calcium channels. Right. What was your name? Alvaro. So, yeah, Alvaro knew the answer. So basically they go for alpha to delta side of VSCC. So that is how they work in the brain. And basically they go for that specific site when the site is open, and they prevent release of calcium inside the cells. And that is the same similar mechanism when we have been talking about glutamate neurons, right? And basically it just suppresses some of the neuronal activities because of blocking calcium, voltage sensitive calcium channels, right? So that is the mechanism that they could basically suppress that sort of higher activity of glutamate that was just pushing the brain system. There are other anticonvulsants that people tried, lamotrigine, carbamazepine, topiramate. I can imagine people will be discussing about that, so I'm not going to go to the details. But the other idea, so we have been discussing about the balance and how the idea of the balance could inform medications, right? So that is the hope that we have in terms of that mechanistic understanding helps us to decide about medications. The other aspect that would be important for alcohol withdrawal syndrome is when we start them with basically treatment, just making sure that the brain would not get toxic. And one mechanism of toxicity is glutamate toxicity. And there is a second mechanism of toxicity in the brain during alcohol withdrawal syndrome, that could basically make lots of negative impact in the brain. What is that mechanism? So a specific neurotoxicity in the brain that could be happening during alcohol withdrawal syndrome, other than glutamate toxicity. So we talk about Wernicke encephalopathy. And what is the triad of Wernicke encephalopathy? What is the triad? Ophthalmoplegia, what else? Confusion, what else? The third one. You are getting very close. So basically, ophthalmoplegia, ataxia, and confusion. So these are the three triads. And why Wernicke encephalopathy happens? We know that these people are depleted from thiamine. And because they are depleted from thiamine, if you give them glucose, you give them kind of source of energy, they need thiamine. Because they do not have thiamine, they would make lots of toxin inside their brain. And then we have specific areas in the brain, and the first area that will be affected is called mammillary body. So that is the area that you can see here, the blue area. It's called mammillary body. It's just above and attached to hypothalamus. Okay? So that area, mammillary body, could be affected because of that. And also thalamus. Thalamus, these two areas are the most sensitive areas. And as you can see here, the light purple one, that is thalamus. As you can see, there are kind of white matter abnormalities and kind of cytotoxic edema on those parts of the brain, okay? So just an interim take-home note about the discussion that we had so far. So we have been discussing about central role of dopamine. We have been discussing about glutamate, GABA, norepinephrine, and withdrawal. We have been discussing about GABA-glutamate balance. We have been discussing about importance of opioid system in alcohol use. We have been discussing about glutamate toxicity. We have been discussing about benzodiazepine as a gold standard for alcohol withdrawal syndrome. We have been discussing about gabapentin, anticonvulsant, memantine, other things that could be important in this process. We have been discussing about the role of anticonvulsants. We have been discussing about metabolic toxicity and neuroscience-informed addiction psychiatry, okay? So I'm going to stop here and make sure that I will give my colleagues enough time to present kind of their discussions. I have another half of my kind of talk discussing about basically I discussed so far about the first and second and third topic to glutamate toxicity. But we have another dimension we call dopamine toxicity that we definitely need to discuss about, and also new psychoactive substances and how they work inside the brain, how buprenorphine is going to work with fentanyl and other hypsos that we have, potential biotyping, and also new molecular targets for medication development, and the new ideas for addiction treatment that we can further discuss about in the second part. Thank you very much for your attention.